Hi, physiology students. Welcome to one of the final two chapters that we'll get through. So this is chapter 23. I've condensed it into one PowerPoint. It's on cellular respiration and metabolism that we'll talk about. And then chapter 24 that I'll post the recording to later will be on the urinary system. And that'll take us through module 4. So I'm going to use the words cellular respiration and cellular metabolism interchangeably here. Metabolism itself is a summary of all the mechanical processes basically used to turn glucose into energy. And we talk about metabolism as a series of anabolism or catabolism reactions. And you should know well the differences between anabolism and catabolism. Anabolism is the synthesis of large molecules from smaller ones, like putting together proteins from their individual amino acid parts. And catabolism is the hydrolysis or breakdown of complex structures to some simpler ones, such as breaking down proteins into amino acids. We call it hydrolysis because in order to break apart bonds between amino acids, for example, we have to take away atoms from that bond, and that's called hydrolysis. Cellular respiration then is a catabolic reaction, so you should know that. So again, cellular respiration is a catabolic reaction, meaning that we are breaking something large into something small. So in this case, we're taking glucose to form ATP. So it's a catabolic breakdown of food fuels like glucose, where energy from food is captured to form ATP in cells. And the goal of cellular respiration is to trap chemical energy in ATP. Energy can then be stored in glycogen and fats, which can be broken down later. Phosphorylation refers to enzymes that shift high energy phosphate groups of ATP to other molecules, and phosphorylated molecules will then become activated to perform specific cellular functions. So these are the 3 stages in processing nutrients. So this is kind of a piggyback off of our digestive system talk. Stage 1 is the digestion, absorption, and transport to tissues. We talked about that in our digestive system. Stage 1 is cellular processing, which occurs in the cytoplasm. This is the synthesis of lipids, proteins, and glycogen, or the catabolism, glycolysis, the breakdown of glucose into pyruvic acid and acetyl-CoA. And then the third step is the oxidative breakdown of other intermediates to form carbon dioxide, water as waste products, but ultimately ATP, which is what we want to get out of cellular respiration. All of our body cells need ATP to function, and this stage 3 occurs in the mitochondria. Cellular respiration consists of glycolysis in stage 2 and all of stage 3, and that's what we'll be going over in general. These are the 3 stages of metabolism of energy-containing nutrients. So basically, proteins are broken down into amino acids, which can enter one of these stages to be kind of shuffled through the cellular respiration process. Carbohydrates get broken down into glucose or stored as glycogen, and fats get broken down into triglycerides. And you can see kind of how each breakdown of these 3 different food groups will then enter this cycle to ultimately create ATP at the end. We have anabolic and catabolic reactions. Remember, an anabolic reaction is creating something larger, and a catabolic reaction is breaking something down. So we talk a little bit about oxidative and oxidative reduction reactions. An oxidation reaction involves the gain of oxygen or a loss of a hydrogen atom, and an oxidation-reduction reaction or a redox reaction will oxidize substances. Oxidized substances will lose electrons and energy, and reduced substances will gain electrons and energy. These redox reactions will occur throughout our cellular respiration cycle. You don't need to know specifically, but you have different kind of enzymes which catalyze these processes. A dehydrogenase will remove a hydrogen atom, and an oxidase will transfer an oxygen. These are 2 main important coenzymes that act as hydrogen or electron acceptors in the oxidative pathway. And these 2 will be really important for accepting excess hydrogen or excess electrons. They will carry these excess hydrogens and electrons to the final step of cellular respiration called the electron transport chain. And I'm going to take a moment here to highlight there is a workshop on cellular respiration metabolism that I'd encourage you guys to go to or watch the video. because it'll help explain this in even more detail, but just in a little bit simpler form. Speaking of workshops, you have till this Friday, or Saturday, I should say, December 11th, to turn those in. So this is just an example showing how FDA at this point is an oxidized molecule. And then when we add two hydrogens to it, we're reducing it, because when we add a hydrogen, we have to add electrons to it, which have a negative charge. So FADH now is a reduced molecule. So this is called kind of a reduction reaction, and that reduced FADH2 will go on and bring its electrons and hydrogens to the final step of cellular respiration called the electron transport chain. So ATP synthesis, there are two mechanisms used to make ATP from captured energy that is freed or liberated during cellular respiration. Substrate level phosphorylation is when we transfer phosphate groups from phosphorylated substrates to ADP. So basically what we do is we have adenosine diphosphate with two phosphate groups. We add on another phosphate group and we create ATP. So that's important to know. To make ATP, we are basically adding an extra phosphate group onto ADP. This will occur twice in glycolysis and once in the Krebs cycle, and necessary enzymes are in the cytosol for glycolysis, so outside of the mitochondria, and in the mitochondria for the Krebs cycle. So this is a look at substrate level phosphorylation. Basically, we're taking an extra phosphate group, adding it to ADP and creating ATP, which again is adenosine triphosphate, and that is what our body needs to perform all the functions. Oxidative phosphorylation, that is a more complex process, but it produces the most ATP, and this will occur in the electron transport chain in the inner mitochondrial membrane. It's a chemoosmotic process where it couples the movement of substances across a membrane to create a chemical reaction. And this is what the electron transport chain will do. Energy will be released from oxidation of food. It's used to pump hydrogen ions across an inner mitochondrial membrane, creating a hydrogen concentration gradient, so that when hydrogen flows back through a protein carrier called ATP synthase, this energy from that flow is used to phosphorylate ADP. But again, phosphorylating ADP just means adding an extra phosphate group to adenosine diphosphate to create ATP. And in oxidative phosphorylation, in the presence of oxygen, we can produce the most ATP, and this occurs in your electron transport chain, as shown here. Simply put, we're taking energy from food to pump hydrogen atoms across, so we get a greater concentration of hydrogen up here. Then the hydrogen rushes through this ATP synthase. So we have phosphorylation ADP plus the phosphate group to create the most ATP. And we'll go through that when we get to electron transport chain. When glucose enters a cell, it's phosphorylated, or two phosphate groups are added to it to create glucose 6-phosphate, and then it kind of moves on and goes through the process of glycolysis. Most cells lack the enzymes for the reversal of this reaction, so glucose usually becomes trapped inside the cell, and only cells in your intestine, kidney, and liver can reverse the reaction to release glucose. So this keeps the intracellular glucose concentration low, which always ensures continued glucose energy, so that glucose is constantly entering your cells to enter this pathway to create ATP. Here's the oxidation of glucose. So glucose is catabolized via the following reaction. And again, that means it's broken down. We have C6H12O6 plus oxygen, hydrogen, oxygen and water. This basically should be an arrow, not a plus sign, to create carbon dioxide, 32 ATP, and heat. So really simply put, glucose catabolism, or the breakdown of glucose into ATP, requires these three pathways. And these are the three pathways that I want you to know well. This is cellular respiration, taking glucose and turning it into ATP. We get about 32 ATP produced from one molecule of glucose, and glucose will enter glycolysis, which occurs in the cytosol of the cells. Then the products of glycolysis will enter the Krebs cycle, and then the electron transport chain and oxidative phosphorylation. On these three steps of cellular respiration, we're going to go over now. You should know this picture and know where each of these three steps occurs. So glycolysis occurs in the cytosol, This basically takes glucose and turns it into pyruvic acid. So we're just kind of breaking apart glucose to turn it into pyruvic acid. Pyruvic acid then enters the citric acid cycle, which is in the mitochondria. These two steps, glycolysis and citric acid cycle, are what we call substrate level phosphorylation. And then the products of the citric acid cycle go on into the electron transport chain. And the most ATP is produced in the electron transport chain via oxidative phosphorylation. So glycolysis is a 10-step pathway. It's the glycolytic pathway. It occurs in the cytosol. We won't go, you don't need to know every step perfectly, but you should know that glucose is broken down into 2 pyruvic acid molecules. The 3 major phases of glycolysis are shown here. We activate the sugar. We need ATP to start this process. So we kind of start the process of activating the sugar by adding into ATP, because those 2 phosphate groups from those 2 ATP will activate glucose and be added on either side of the glucose molecule itself. And you can see here that we are in the cytosol of the cell. We're outside the mitochondria at this point. The sugar cleavage, then that sugar, the fructose 1,6-biphosphate, is split into 2 3-carbon fragments as shown here. So this is kind of the next phase of glycolysis. We're basically just splitting that sugar in half. And then we have 6 steps involved in sugar oxidation and ATP formation, but the end result is 2 pyruvic acids that are formed and 4 ATPs that are formed by substrate level phosphorylation. These 4 ATPs are formed. We say net total gain of ATP from glycolysis is only 2, and that's because we require 2 to start the process. So even though we're creating 4 ATP at the end of glycolysis, since we started, we had to use 2 to start the process. Net ATP total for glycolysis is only 2. Then here we have then finally the final stage. We're taking phase 3. We're basically through substrate level phosphorylation creating 4 ATP, 2 pyruvic acid molecules. And these pyruvic acid molecules will then go on into the citric acid cycle, which occurs in the mitochondria. This occurs in the presence of oxygen. In the absence of oxygen, pyruvic acid will actually be changed into lactic acid, and that will build up in your muscles and cause a burning sensation. So here we have 3 major phases of glycolysis as shown here that we went through individually. Again, what you need to know, glycolysis takes glucose and basically splits it apart, turns it into 2 pyruvic acids that go onto the citric acid cycle. This occurs in the cytosol of the cell, and then we'll move on to the next step. So this just explains how there's a net gain of only 2 ATP, because even though we are producing 4, 2 ATP are subtracted from that first phase. All right, and then this describes how if no oxygen is present, the pyruvic acid gets reduced to lactic acid. Otherwise, if the oxygen is present, the NADH will transfer its hydrogen to proteins, and we can kind of continue the cycle in the electron transport chain and Krebs cycle occurring in the mitochondria. So we'll kind of keep going here. I don't talk too much about lactic acid, but it can build up and cause problems if that occurs. So the citric acid cycle, this is the second step of cellular respiration. This occurs also known as the Krebs cycle. It occurs in the mitochondrial matrix. It uses the pyruvic acid from glycolysis. Pyruvic acid must be actively transported into the mitochondria. Once inside the mitochondria, pyruvic acid begins with what we call or enters into a transitional phase. This transitional phase has 3 steps, but all it's doing is it's basically changing pyruvic acid, and it's being converted into acetyl-CoA, which will then go on to enter this cycle process. So here we have the transition phase. We're taking the pyruvic acid from glycolysis and turning it into acetyl-CoA. And then acetyl-CoA, remember we have 2 pyruvic acids, so we'll have 2 acetyl-CoAs. So this cycle will go around once for every acetyl-CoA molecule. So it goes around twice. And basically what's happening here is acetyl-CoA gets oxidized. We have carbon dioxide given off as waste products. We have the formation of our high energy electron carriers, which is NADH. and FADH. And the formation of these is really important because they will take their electrons to the last step of cellular respiration called the electron transport chain, which is needed to produce the most ATP. So we're producing a little bit of ATP in the Krebs cycle through substrate level phosphorylation, but the majority and the most important thing we're doing is we're getting rid of CO2 as a waste product. And our bodies get rid of CO2 when we exhale that carbon dioxide. And then we're creating these high energy electron carriers, the NADH and the FAD. So here's our citric acid cycle. It will go through this cycle. I explained this a lot in the workshop video. So I'd really encourage you to watch that workshop video. I don't ask you too many specifics about this on your exam, but just in general, you should know that the citric acid cycle basically is taking that is acetyl-CoA and then producing different carbon molecules. For each turn of the cycle, it goes around twice, one for each acetyl-CoA that enters. We get 2 carbon dioxide, 4 molecules of those reduced enzymes, which have our electrons that we need, and only 1 ATP. So if we add the total final products, and you don't need to know total final products throughout, but we're not at too much ATP yet, and that's our goal for this. So I'm going to kind of go over this, and then the third step is the electron transport chain and oxidative phosphorylation. So oxidative phosphorylation is where the magic happens, where we're producing the most ATP in the presence of oxygen, and it's divided into 2 phases, the electron transport chain, which creates a proton gradient across the mitochondrial membrane using high energy electrons removed from hydrogen, from glucose, and chemoosmosis, which uses the energy of the proton gradient to synthesize ATP. This oxidative phosphorylation, these 2 phases occur in the inner mitochondrial membrane, and this is shown here. So here's our inner mitochondrial membrane. Remember, our mitochondria has 2 membranes to it. You can kind of see the outer one up here where my laser is. And then here's the inner mitochondrial membrane. We have a series of these proton carriers, which are going to take the electrons and the hydrogen atoms from these reduced coenzymes. They will deliver their electrons to these complexes. Those electrons will be pumped through the complexes to basically pump hydrogen ions out into this intermitochondrial space, producing the proton or hydrogen ion gradient. When they do that, so the electrons will be transferred from one complex to the next. Each complex is reduced and oxidized. The energy released will pump hydrogen ions into this intermembrane space, creating an electrochemical gradient. Coenzyme Q is ubiquinone. This is just, and cytochrome C. These are they help to shuttle electrons between the larger complexes. At the respiratory complex 4, 2 electrons combine, as shown here, with protons and half a molecule of oxygen forming water. And this is why it's so important for your cells to need oxygen, because what oxygen does is it combines with these excess electrons to form water as a waste product. Then finally, so that's phase 1, transporting these electrons to create this proton gradient. And then phase 2, chemoosmosis, uses this proton gradient to synthesize ATP. So we have little rotor machines that turn when hydrogen gets pumped through them or passes through them. And these little rotors turn an ATP synthase machine. And this is where oxidative phosphorylation occurs. We take ADP, we add a phosphate group to create ATP. And this is where the majority of ATP is formed in cellular respiration. So the electron transport chain is the only pathway that directly uses oxygen. And here's an overview. Basically, the electron transport chain uses energy from those electrons and hydrogen to pump hydrogen across the membrane, and excess hydrogen and electrons will combine with oxygen to produce water. So oxygen is called our final electron acceptor because it gets rid of excess hydrogen and electrons. Here is a little bit more about the electron transport chain, the main steps involved in it. And then finally, at complex 4, you should know this is where electrons pair and combine with some hydrogens and a molecule of oxygen to form water. And then we kind of have this series of reactions in the electron transport chain to form water. And then the chemoosmosis is where the hydrogen re-enter the matrix only via this ATP synthase, which is like a small rotary motor that drives the addition of that phosphate group to ATP, creating our most ATP present. So this describes the chemoosmosis, how the hydrogen now flows down its concentration gradient through this motor. The motor spins and forms ATP. It's really interesting how the motor spins, and you can read through the steps here, and it's even more fun to see it underneath a microscope. So what you're looking at is all of these little ATP synthase molecules, these little motor or rotors that will spin as hydrogen travels through them. And as the hydrogen travels through, we combine a phosphate to an ADP to create ATP. And it's actually fun to see those little rotor motors on an actual picture. Some homeostatic imbalances. So we have some toxins that actually affect the electron transport chain. So example, the gas used in gas chambers, cyanide, will disrupt oxidative phosphorylation by kind of binding or blocking the cytochrome oxidase. This will block electron flow. And if we don't have electron flow through the electron transport chain, we cannot have this rotor creating a bunch of ATP for us. So your cells will die if any part of the electron transport chain is disrupted. And cyanide and other poisons, some poisons will abolish the proton gradient, so no ATP will be made. So a lot of toxins and poisons kind of interrupt this process. And again, death occurs almost instantly because if your cells are not creating ATP, they will die off. So here's some summary of ATP production. It's a very efficient process. Basically, simply put, ATP cellular respiration is about 38% efficiency, meaning how it uses the fuel and what's lost as heat. And in summary, even the best machines that we can make with human hands are only about 10 to 30% efficient. So substrate level phosphorylation produces about 4, oxidative phosphorylation produces about 28. So in general, we're producing about 30 or 32 ATP for just one molecule of glucose, remember? And that is still a little uncertain. Different textbooks will tell you different things. So anywhere between 28 and 32 will be the total amount of ATP produced for each molecule of glucose. And this is, again, a summary of the energy yield during cellular respiration, the 3 steps: glycolysis, producing pyruvic acid being turned into acetyl-CoA to enter the citric acid cycle. This step is sometimes called the transition phase, but it also occurs in the mitochondria, and then the electron transport chain and oxidative phosphorylation. So for your lecture, you should know really simply those 3 steps, what metabolism means, catabolism, anabolism, these 3 steps of cellular respiration, because there will be several questions about those. And then that's the end for this chapter, but I will see you guys. We'll try to upload the next chapter, chapter 24, which is urinary system, by tomorrow. So take care, guys, and thank you for listening.